Dr. Platt got started in his scientific career when he graduated Morehouse in 2001 with a Bachelor in Science in Biology. But then he kind of moved over to the brighter side of biomedical engineering. He joined this department at Georgia Tech worked in Dr. Hunjung Jo's lab over at Emory. During his work, he received the NASA Jenkins Fellowship and conducted thesis work on flow-mediated mechanisms of proteolytic remodeling in atherosclerosis. He graduated in 2006. Upon graduation, he began work at postdoctoral work in joint between Linda Griffith's lab and Doug Laufenberger's lab at MIT, working on orthopedic tissue engineering and systems biology. He completed that work in just before 2009. As he was finishing up his postdoc, he was also simultaneously trying to set up his lab here, which opened officially in January of 2009. I'll let Dr. Platt talk more about what our lab does, obviously, but it's pretty much a fusion between his thesis work of tissue remodeling and his postdoctoral work of systems biology. Over the course of these, I guess it was 2 3/4 years we've decided that we've been here, he's already kind of gotten a pretty prestigious list of awards, the least of which aren't the NIH Director's New Innovator Award. He's also a Georgia Cancer Coalition's Distinguished Cancer Scientist. And he's also gotten the Creative and Novel Ideas in HIV Research Award from the NIH and the International AIDS Society. Additionally, he's also going to become kind of a world traveler at international conferences, going to Italy, he's been to South Africa, and he's been to Spain this past spring. So it's been quite a whirlwind adventure getting this whole lab started. So I'm very interested to see how he ties all the research in our lab together.
Thank you. Thank you very much, Phil. Yeah, Phil walked in the door with me. I walked in on the payroll, Phil didn't, but I still appreciate that he was able to stick with us. So I love the Breakfast Club, so it's a great opportunity again for us to talk to our Georgia Tech people. And for those that don't know, I always have to plug the GAP seminars, which is kind of like the Breakfast Club for our grad students and postdocs, for them to present their work every Wednesday at 12, so make sure you come on out. And so today I've always seen some very fun Breakfast Club seminar titles, so I thought I'd come up with one of my own. And so what's eating you? We study proteases in my lab and really in the context of tissue remodeling. And as Phil said, we actually do lots of different things. I'm going to attempt to tie them all together. For those in my lab that if I don't spend as much time on your projects as you would like, it's only because they only give me 50 minutes, okay? So here we are with the Platt Lab, Repair, Regeneration, and Remodeling. And again, we really focus a lot on cardiovascular tissue remodeling in 2 main diseases, sickle cell disease and also an HIV-mediated atherosclerosis. And again, this harkens back to my PhD work in dealing with disturbed flow and plaque remodeling. But we also spend a lot of time with bone marrow-derived stem cells, which I'll speak just about briefly. And then, of course, we're engineers, so to prove that we're not just doing biology, we also throw in some computational analysis, and that brings in some math modeling plus some of my systems biology experience. So what is tissue remodeling? I like to think of tissue remodeling as just how you go from this healthy vessel to this disease, not vessel, but healthy structure to some disease structure. So this is atherosclerosis developing. This is dilated cardiomyopathy. You can see this thickened heart wall. Emphysema, you get degradation of that alveolar tissue. And then, of course, in osteoporosis, you start to break down this bone mineral. And usually these involve some amount of protein degradation followed by protein synthesis and also cell infiltration. And as you can imagine from engineers, once you start to change the properties or this tissue composition, you start to change the properties. And so one of the first things that was kind of beat into us when I started this graduate program, the second class, which again still is the best class ever, is that structure relates to function. And so I like to analogize it with looking at this medieval castle, and it looks, looks very different than how houses look today, but it wasn't built this way just to look this way. So these turrets at the top are very much so that you can have bowmen and archers kind of peek out, shoot their arrows, and then hide behind them for protection. The small windows and slits of windows, they didn't have central air conditioning back in the day, so they didn't want to lose heat or take in too much of the, for temperature regulation. Versus this beautiful condominium building in Miami, this is built for luxury and to make your neighbors jealous. But again, the structure of both of these buildings relates to the function. And this is also very much true for biology and physiology. So here we have the small intestines and the large intestines. And again, they're not just folded up inside just because someone said that's going to be a great way to, or they just look pretty this way. It's actually that your small intestines, as many of you know nowadays, they stretch way across this room. but they have to be all convoluted and folded to increase surface area to bring in nutrients from the food you've eaten. Even better, on the inner surface are these microvilli, these finger-like projections. And again, not just to have fingers in the intestine, but each of these projections also increases the surface area for maximal diffusion of nutrients or even active transport. And then I love to look at the ear. I don't study the ear, but it's just this amazing architecturally designed organ that you can amplify small vibrations and sound waves and amplify them and turn them into biochemical signals. So again, the structure relates to the function. Now what we care about is when you mess up the structure, you mess up the function. So for example, your nose is very much to breathe in fresh air and oxygen. When you close it off, you get brain damage. And so these are the kind of things that we kind of look at. How when you change the structure, does that function change? So I'm not going to spend a lot of time on bone marrow, but I love to talk about it because, again, I got really into this in my postdoctoral experience. But the bone marrow is this amazing home to progenitors, one could call it, stem cells and other progenitor cells. In my group, we actually do care a lot about MSCs and HSCs, and we'll talk a little bit about these today. But some other projects that are happening in my lab in the background we're also looking at breast and prostate cancer metastasis and how they then home to the bone marrow. Because once these things get to the bone marrow, that's actually a very poor prognosis for survival. And so then the bone marrow does become a home to cancer. And so Gerald Dumas, a postdoc, and Caterwald, a grad student, are actually starting to look at how when these cells enter the bone marrow, how do they interact with the MSCs and the osteoclasts to actually promote either further cancer growth or further, increasing the size of these tumors, and then how are these signals cross-talking with each other to promote or hinder that. So we can come up with some ways that we can kind of slow this progression, because right now it's, there's not a lot of options. But the real thing that we love in these bone marrow cells is these cells that get out, these hematopoietic stem cells. And these hematopoietic stem cells, they differentiate into all of the blood formings, all the blood cells that you have in your body. So this one cell becomes so many different types of cells. So you can imagine the amount of proliferation, differentiation that has to happen. But what I'm going to spend a lot of time on is they become monocytes. And one project that Kun-Young Park in my lab is working on is these monocytes are not terminally differentiated cells. And so again, we like to think of these as progenitor cells, because as they circulate in the body, they then leave and enter different tissues where they can then differentiate into macrophages and osteoclasts. And again, hematopoietic stem cells are also going to be very important when I enter or begin the discussion on our work in sickle cell disease, which is mutations in hemoglobin, which is a protein found in your red blood cells that differentiates from HSCs. But first, why do we care so much about macrophages and osteoclasts? Well, or even monocytes. So monocytes are something because, again, they circulate, but then they receive danger signals telling them to enter tissue. So when they enter into arteries, it's one of the first steps in atherosclerotic development. They then differentiate into macrophages, which then pump out proteases to help remodel this arterial wall. And again, we're thinking about things in the context of remodeling. They also are infectable by HIV, and so one of the projects I'll discuss also is how we care about why do people with HIV have increased cardiovascular risk. So our hypothesis is there's something about these infected monocytes that differentiate into macrophages that play a part. Another big thing that I just want to mention too, in case I don't get to it later, is the drugs are very good now to protect people from HIV and lower viral loads, but as we all know, when you go off the drugs, the virus rebounds immediately. And it's also believed that macrophages are these harbingers of these viral reservoirs that when you remove the drug pressure, then the macrophages release the virus and they come back. So these actually are playing quite a bit of role in many different diseases. Another one that's studied very heavily here in the Yoga Nathan and the Joe lab, cardiovascular calcification, whether it be in the heart valves or the arteries. Monocytes can also enter the arterial wall and, depending on the cues they receive, become macrophages, but there's also a lot of evidence they differentiate into osteoclasts in your arterial wall, which normally should be in your bone. And so, again, monocytes are playing many different roles. So I like to think of it as this tale of two niche, right? So macrophages, what are they? They are helpful cells. And they help in different inflammatory environments, and they also phagocytose bacteria and invaders. But they also become associated with tumors and also play a part in tumor progression. Osteoclasts, again, they really are in this bone marrow and serve a major role in calcium homeostasis to resorb bone and release calcium for cell signaling everywhere else. But when overactivated, they also are indicated for osteoporosis. So again, there seems to be this balance, but this niche or where these cells go helps determine what final phenotype these cells will become, and then what will they do. And so why do I care about these two cell types? Of course, because they pump out my babies. These are my favorite enzymes in the world. The lysosomal cysteine proteases, commonly called the cathepsins. And so I got to work with these during my PhD got started. But they were first identified in lysosomes. But again, they've been shown to be secreted by osteoclasts, these bone-resorbing cells, macrophages, and many other cell types. And really in many other cell types only under disease conditions, okay? What's really important about them is they are the most potent mammalian collagenases and elastases. So for example, you'll hear me say Cathepsin K a lot. It can degrade and hydrolyze type 1 collagen, strongest tensile protein in our body, much better than any other human collagenase that we have, okay? And Cathepsin V is actually the most powerful elastase that we have in our bodies. So why they become so controversial and what I like to say is very much understudied is because, again, they were first identified in lysosomes because they do prefer an acidic environment for their optimal activity, okay? And they also prefer this reducing environment due to the cysteine at the active site. And so the common thought used to be, oh, well, when they get outside of the body, they won't work because it's neutral outside of the body. And as we know now, there's many different inflammatory diseases and other diseases that show that there's local, at least, acidification of at least the immediate microenvironment that can aid these enzymes in doing their work. Another trick is that they are synthesized as an inactive precursor, as this zymogen, that has to be proteolytically cleaved to its active form. The precursor is very stable at neutral pH. The active form is not, and so there's been some tricks and confusion there, which we'll discuss. But again, as people started to open their mind into what these things can be a part of, many of your favorite tissue-destructive diseases have been implicated with cathepsin activity. And so again, my enrollment my initiation started in Dr. Joe's lab, where people at Georgia Tech have done all this major seminal work on how changes in blood flow patterns actually induces atherosclerotic plaque formation. And so during my dissertation, we published a series of papers that show how just shear stress and fluid flow actually can upregulate these enzymes. So where you see the disturbed flow and plaque formation is where you see upregulation of the cathepsins. So again, what made it difficult to study during my dissertation? I tried to remove that burden from my graduate students, and they still don't appreciate it. But we've been making progress because some of these burdens have been lifted. So here's the challenge. Cathepsin K, very stable at neutral pH. This is how it's synthesized. Under normal conditions, it's synthesized, sorted through the Golgi, and targeted to the lysosomes, should never escape the cell, unless it's an osteoclast, which will specifically pump it out. But under times of disease, when their upregulation is amplified, they get mistargeted and missorted and end up being secreted outside of the cell. Secreted as this inactive proform, and you can see this pro piece here that blocks the active site so that the enzyme cannot work. Low pH, this enzyme can autocatalytically cleave itself to activate it, but also other proteases can activate it from the pro to the mature form. And you can see there's a reduction in size because you're going to lose a bit of this propeptide, and you get this mature enzyme. Now the mature enzyme is now able to bind substrate, hydrolyze it, and do all the dirty work. But if that enzyme is, again, in the pro form, it does not bind substrate, or if that enzyme has been denatured or degraded, it will not bind substrate and do any work, okay? Even if it's still present. So there have been challenges in the field, which I always write at the beginning of every grant that I write about what's been difficult with detecting them. But here are the big issues. Under acidic pH, they're more stable. So neutral pH, they still have activity, but they stay around shorter. The proform of the enzyme stays around a lot longer, and most antibodies detect the proform and the mature form. And then the substrates that they hydrolyze are very promiscuous among several cathepsin family members and then several other proteases. So specifically targeting them using a substrate, the wording has changed from selective substrate, or from, they used to call it a specific substrate to now a selective substrate because everything hydrolyzes it. So it's made things a lot challenging. So when I got back to my lab, I wanted to remove a lot of those challenges. So let's look at what we know. We can detect the proform of the enzyme because it stays around quite long. And we can also detect products of the Cathepsin K hydrolysis. But how do we actually detect this mature form that doesn't stay around for a very long time, and its activity, according to what used to be done, was actually quite in a high amount of signal to noise. Low amount of signal to noise, excuse me. So we're engineers. So the first thing we could do is design and solve a kinetic-based enzymatic math model. The amount and activity of the mature cathepsin can then be back calculated from these other things that we can determine. And then part two, which we'll talk about, is the development of these sensitive assays to improve its detection. So when I think about this computational kinetic model development, again, now that we're starting to see there are so many more cell types than just the ones initially thought. So these enzymes could be released if you was in some epithelial cell, like in a cancer environment, could be released apically or basally. Macrophages, mesenchymal, they can release it at all sides. So we're just trying to consider really if it's, what happens when it gets released, and then whether it be in either of these spaces. I always spend too much time on this slide because I break it down on the next one. There we go. So if we were to look at a single cathepsin, they are most often released as this inactive proform. So you have some proenzyme released, and there's some rate at which it gets activated to the mature enzyme. At that point, it can either be bound to the inhibitor. There's a protein inhibitor that's present in all of your extracellular fluid that binds to them very tightly if it finds it. Or they can bind to substrate. And of course, when they bind substrate, they can either hydrolyze it or they can release it. But either way, you return back to the mature enzyme, which has some rate of degradation based on the conditions and the space in which it's surrounded. So a lot of biochemist studies have been great at identifying these rates of the catalysis of the substrates, but the context-dependent rates, such as the activation and the rate of degradation, those depend on the situation, the organ, the tissue that you're looking at. And so these have to be determined independently. And so again, you can develop your governing equations, solve the systems simultaneously, and then fit your parameters to fit the activation and the degradation. And Zach Berry, an undergrad in my lab, does this for us all the time, which is great. And so we use this substrate as just to make sure we could do it. This is one of the formerly identified specific substrates for Cathepsin K, which now lots of different things degrade. But if you have just Cathepsin K in the system, you can get a readout for its activity. And we're going to expand on this model later, but I'll come back to that. Because if any of you have ever seen my students give a talk, or seen me before, of course we love our favorite assay, and yes, you're going to see the schematic again. Because we wanted to identify specifically these enzymes. So we developed this method of Cathepsin zymography, and a lot of people that have done MMPs have done zymography. And I'll describe this process, but it had not been worked out for Cathepsin, and I'll tell you what some of those challenges were. So what is zymography? Everyone runs SDS Page or does Western blot. So we make a polyacrylamide gel just like you would for SDS Page, except we impregnate that gel with gelatin, which is denatured collagen and a substrate that many proteases hydrolyze. So then when we lyse the cells or use isolated enzyme, we can then load that into this loading buffer, which is just like your Western blot, except this does not have beta-mercaptoethanol. So we're not breaking any disulfide bonds, okay? And that's going to be important for later. I load in the gel, and we can apply electrophoresis, separate the proteins by their molecular weight. Now this is important because many of these enzymes actually, they're close between the 25 and 30 kilodalton range, okay? But when you separate them by their molecular weight, you also take advantage of, because we don't reduce any of these bonds, each of these has about 5 to 7 disulfide bonds. They also now have a different hydrodynamic radius, and so they will migrate differently through the gel than you might perceive them to in a Western blot. Or in a reduced gel, let me say. So instead of transferring them to a membrane, though, we transfer them to this renaturing buffer. Now remember, we added SDS, which has a negative charge, so it's going to partially denature or linearize these proteins, but only so far as those disulfide bonds, hold them together. So if we wash out the SDS, these enzymes actually refold inside of the gel, and importantly, at the molecular weight or the electrophoretic distance where they migrated. Then we can transfer them to this assay buffer that has everything perfect for their activity. We do it at pH 6, we give them the DTT for reducing environment and incubate them at 37 degrees overnight, where these now refolded enzymes can start to hydrolyze that gelatin substrate in the gel at the size where they were separated. Such that the next morning we can stain with Kumasi blue. Because there's gelatin in the whole thing, everything turns blue except where the enzymes have eaten away, and you get these cleared white bands of activity. And so when we first got this work, we were very excited, because you can imagine, because it hadn't been shown for Cathepsin K before. And you can see here we activated pro-Cathepsin K to the mature K, and again, you see the shift in the band. This is by Western blot, which gives you the amount present. Then for the zymography, we use aliquots of these same samples, and you can see here we were getting bands in both the pro form and the mature form. And if you remember, I said the pro form is not active, but we're still at this bottom molecular weight. And what this actually led us to think is, wow, maybe there's some mature K already in the pro aliquot, and we couldn't pick it up by blot, but the zymo is much more sensitive. And so we went back, we also did the model, I'm not going to show that for the sake of time, but turns out that our zymo is actually much more sensitive than the Western blotting. And this really is the best Cathepsin K antibody they have on the market so far. And so we're able to detect femtomole quantities of mature Cathepsin K using our zymography. So we were like, great. Now let's start to use it on things that we find important. Oh, so this is the first paper we got out, and we were excited. So then what about other Cathepsin family members? Because many of them are important, not just K. We just love K for many different reasons. But can we multiplex the assay and the model? And so Katera Wilder came, she was a summer student here first, and then she's now a second-year graduate student here. But she began to work out how do we multiplex this Cathepsin zymography. And we've actually been able to come up with a protocol where we can detect Cathepsins K, L, S, and V. And you can see they all run at different molecular weights, I'm sorry. These are pure enzymes isolated from E. coli, so there's some glycosylation issues here. And then these are actually from mammalian expression systems. And what we love is that they all, again, do run at different molecular weights. This is K, L, S, and V, and she loaded them all together, and you can see how they just translate across. And we validated them with Western blots to show their identity. Now again, the part that's interesting here too is you see that for Cathepsin L and Cathepsin V, they actually produce several bands by Western blot, but there's only the one that's giving us the zymographically active signal. So that's going to be important when we actually start to consider how much of the actual enzyme are we getting, but we can still see relative changes. And we love it because we could do it all from one cell lysate, which I guess was the next part. Can we assay it, and then can we apply it to cells and tissue? So we made the tools. Again, the model is in the background. I'll come back to that later. But now let's talk about the diseases, because we don't want to just make tools just to make tools. We want to then use it to apply them to human health and disease. And so I bring you to sickle cell disease, which is a major thrust in our lab. And it was one of these things that when I started my independent position, surprised me how much work was not done on it. So for those that don't know, sickle cell just last year celebrated its 100th anniversary. Not celebrated, but 100th anniversary. The first patient was diagnosed or identified with it in 1910 in Chicago. And it's also the first, genetic disease that's been identified. Single point mutation from this A to a T. Changes this from a glutamic acid to a valine. Glutamic acid is hydrophilic and has a negative charge. Valine is hydrophobic. So the point of this mutation is that once the hemoglobin molecules release the oxygen and they undergo these slight conformational changes, it exposes this hydrophobic valine to the hydrophilic space. Of course, that's thermodynamically unfavorable. So all of the valines then start to cluster together and form these very stiff hemoglobin polymers inside of the red blood cell. And it's this unique disease because, again, it's one point mutation in one amino acid and one protein, hemoglobin, that's only in red blood cells. But you have trillions of red blood cells, and that becomes the problem. And so the other thing that's significant about it is that red blood cells, as we know, do not have a nucleus. And so for those that do biomechanics and cell mechanics, you know, the nucleus provides that rigid structure, and there's nothing here to retard that hemoglobin polymer from really just deforming these red blood cells. And so this causes multifactorial damage. And what most people know about is that it causes these vaso-occlusions that actually start to block blood flow, which of course is very damaging. And I love this picture of these spiculated red blood cells where the hemoglobin polymer doesn't form in just one axis, it can form all throughout. And so this is clearly a problem, and I have this movie that Phil made. We have a hypoxia chamber as well. And so you could see, like, so this one's getting ready to unsickle, and these are going to be changing. And so we had to sickle them first. It's kind of played backwards. We put them in the hypoxia, and then we took them to our microscope and did the movie. But you can see these things, it just all happens by thermodynamics. But what became interesting is, you know, a lot of people were studying in the veins where the low oxygen environments were for these obvious reasons. But in starting to understand what was happening, there's actually 4 different phases of these red blood cells, or 4 different life cycles, where these are the early reticulocytes. They're very deformable, as you can see. And as they get older, they eventually become these irreversibly sickled cells, okay? Because due to this membrane damage, the membrane is completely destroyed, they're dehydrated, and they're just stuck in these conformations. And so there is this idea, it used to be thought that you only had sickling of the red blood cells in the veins. But then you start reading the literature, well wait, you have irreversibly sickled cells. And it's important to also mention, a normal red blood cell stays around in your body for 120 days. And a person with sickle cell disease, they only survive for about 9 to 10 days. So this is why it used to be called sickle cell anemia, because your body is constantly trying to pump out red cells to carry the oxygen. And so when we came along, you know, we were like, well, what are we going to do? We like high flow and these other things. But then it turns out that children with sickle cell disease have an 11% chance of having a stroke by the age of 20. And actually the greatest risk age is age 3 to 5, okay? And this failed so powerfully. This is the brain of a 3-year-old girl. It's terrible though. And so what also happens is they also have silent infarctions or silent strokes where they don't present with paralysis, but these kids, some of them are perceived to have cognitive defects, which were thought to be because of this and that, but then they go into an MRI and they find these silent strokes. And so there's no reason that a 3-year-old or 5-year-old or even a 12-year-old should be having strokes, which are arterial diseases where you have oxygenated blood. So that's where we came along with our crazy idea. But it turns out that luminal narrowing in sickle cell disease is a result of many cell and combinatorial factors. For example, these red blood cells, when they break open, they release hemoglobin, and heme, that iron group, is very toxic. That also scavenges nitric oxide, and for our cardiovascular friends, nitric oxide is important in vasodilation. But if the nitric oxide cannot get to the smooth muscle cells, the vessel never opens in response to shear stress. Additionally, they don't show it here, but there is smooth muscle cell proliferation, which leads us to guess there must be some proteolysis of this elastin layer that keeps the smooth muscles at bay. And all of this works together to have these sticky red blood cells that stick to endothelial cells that then cause these monocytes, the trouble cells again, to begin to stick. And monocytes actually have rigid nuclei. So when they get stuck, they tremendously close off the space with which blood can flow. And then you get thrombotic events and a stroke. But no one knows really some of the factors that's starting all of this. And so these are the things that we wanted to investigate. And so before we got our shear system up and running, we first wanted to see, well, what is just the role of these monocytes? Because again, from atherosclerosis, we know that monocytes are one of these early steps in starting this plaque formation. So how is this interaction between the monocytes and endothelial cells playing a part? And so I'm going to actually describe using peripheral blood mononuclear cells that we isolate from humans, people, and then also we use a monocyte cell line to get at some of the nitty-gritty details. And it's also important to remind you again, in sickle cell disease, these people are under constant damage, constant inflammation, so their circulating TNF alpha levels are actually much higher than a normal person. And TNF alpha activates monocytes to bind to endothelial cells and start the rest of this process. So yeah, I didn't over contrast these, but it makes it look shadowy. So the first thing, this is, I should have had a picture of Phil here, sorry. This is actually Phil's work. He's the one that started a lot of this. So the first thing we did is looked at some of these conditions that are present in sickle cell disease and see if any of these can induce proteolytic activity. So again, there's local hypoxia because these vaso-occlusions cause hypoxia to happen. Also, there's increased TNF, and there's also the release of this reactive or the heme that generates reactive oxygen species. And the first thing we noticed that the overwhelming trend was that anytime we had TNF alpha added to the system, we got increased cathepsin activity. And so you're wondering, how do you know that this is K? So the K actually comes very close to V and the endothelial cells when we test it. And so we ran a few control experiments just to be sure that that was Cathepsin K. So we transfected our endothelial cells with the Cathepsin K plasmid, and you can see it appears right above the V band. And then also when Phil stimulates with TNF alpha, you can see the K appears again. And what Katera figured out was that if you incubate your zymography at pH 4.0, you actually select for Cathepsin V over Cathepsin K, and then you can confirm which band is the K band in other tissues. So we've used both of these to be sure that what we're looking at is Cathepsin K and Cathepsin V for quantification. But it looked like S and L really weren't affected, but Cathepsins K and V. Again, why is it important? K is the most powerful collagenase, V is the most powerful elastase, and both of those are major structures in the arterial wall that provide the biomechanical support. So then we said, okay, well, let's get some blood from people with sickle cell disease, and so we've partnered with the Georgia Sickle Foundation and established a nice relationship with them where we can go and get blood. They can give us blood from homozygous normal individuals and homozygous sickle individuals, or trait. We just place an order, kind of, and then they have to recruit the donors. And so I just like to show this picture. It's a little dim here, I apologize. But there are the PBMCs from the sickle cell individuals are much more adhesive than those from a normal individual, even when stimulated with TNF. And you can see this, Phil counted these cells right here. So again, they're predisposed to have sticky endothelium, sticky cells. And then when we looked at cathepsin activity from these patients, I mean, it was surprising. I mean, with the AA-PBMC, so from homozygous normal individual, you know, you get your V like we saw before. When you stimulate with TNF alpha, you do get this increase in Cathepsin K, but the sickle PBMCs, we didn't even have to exogenously add TNF alpha. Just by taking the PBMCs as they were and incubating with ECs, we saw this significant induction in Cathepsin K, really telling us that these people are predisposed to activate this activity and might be a clue as why they're having such arterial remodeling, even at earlier ages, without the lipid deposition and everything else. So then we wondered, well, what happens if you combine TNF-alpha and monocyte? Oh, so I'm sorry. Now we switched over to the THP1 monocyte cell line so we can start to get at some mechanism. Because there was huge variability in the patients, as you can imagine. There's very much different severities of the disease. So we went to the THP1 monocytes and kind of repeated that experiment and again showed that TNF-alpha alone stimulates cathepsin K and V, but when you incubate it with TNF-alpha and monocytes, you get this increased activity. And again, it seems that TNF-alpha is turning on cathepsin K. It's off at baseline. It's not supposed to be on these systems. And then under disease, it turns up. So we wanted to say, well, they're circulating in the blood. Do they need to bind to increase this activity? And so Phil did a transwell assay where he kept the monocytes separate from the endothelial cells and then cultured them together to determine if we would see that significant increase in cathepsin activity even when separated, like if there was some soluble cytokine that they were secreting. It turns out, yes, when they're in indirect contact, they do increase it, but not at all to the extent that they do under direct contact, okay? So when these cells bind, which they're predisposed to do, they're turning up these proteases. So then we said, well, let's look at what are some of these intracellular signaling mechanisms that are playing a part. And you know, we love Bioplex and the Kemp lab and Linda for helping us out with that. And so we said, well, that's a great way to screen several kinases all at once. And I have this kind of hypothesis I go from that there are these 5 to 7 integrator kinases that kind of maybe can capture most of that information. So we probed the major ones. And what we found was actually quite interesting. ERK is usually involved in everything. I mean, everything. Extracellular signal regulated kinase. And it really wasn't changing in response to monocyte binding or TNF-alpha, where we were seeing the increased activity. But AKT was interesting in that it really responded when monocytes were bound. So whenever monocytes bind, we saw this significant increase in AKT phosphorylation. And JNK and C-Jun were significantly increased in the presence of TNF-alpha. Now remember, that was where we were seeing the most cathepsin K, and C-Jun is actually the downstream target of JNK. So we came up with the new hypothesis, well, if JNK is up and we see increased cathepsin K up, can we inhibit JNK and prevent this increased cathepsin K? And the reason this is actually very important too is because there's no good drugs out there for people with sickle cell disease. There's 100 years. One drug on the market, hydroxyurea. It was an anti-cancer drug that now they use to kind of help stimulate hemoglobin, fetal hemoglobin. But that's it. The only way they help kids that are at risk for strokes, they have to get monthly blood transfusions every month. So we said, well, we can come up with a new drug target. That'd be exciting. And this was amazing. I mean, we incubated with SP600125, a JNK inhibitor. You can see here without it, the ECs alone are not bothered. But when you stimulate with TNF-alpha, you get the increase. And with the inhibitor, you see this reduction. Monocytes didn't really respond. And then again, when you put the two together, you saw this increase in K, and you could block it just by adding this JNK inhibitor. And you can see it quantified below. It actually worked for K and for V, which was quite astounding. I'm sorry, we were quite excited about this result. So then we said, well, okay, that's in our THP1 monocyte cell line. Let's see if it works with blood from people with sickle cell disease, ultimately where it would need to be used. And so we got the blood again, and again repeated that experiment. This is just showing it from the SSPMCs, the same as the AA. But you can see here with TNF-alpha, you stimulate it, you add the JNK, you block it. But here with the SSPMCs, no TNF-alpha stimulation, already you're seeing the increased cathepsin K. But if you add the JNK inhibitor, you can see that we've reduced it. And again, when you stimulate with TNF-alpha and SSPMCs, you get this much higher increase, and you can still get a significant reduction. So this was a great way to show that there may be a way that if you can inhibit JNK, you can at least protect these proteolytic activities that might be exacerbating some of the damage we're seeing. And so this is what was funded for the new innovator, where that's actually from the funding. We didn't have all that data before. But our huge goal there is really to develop these personalized medicine strategies to predict these strokes in children. What they do now is they do a transcranial Doppler and look at velocity in the middle cerebral artery. And the higher that velocity is, once it crosses 200 centimeters per second, they say monthly blood transfusions. But there's no really way to know. Again, so only 11% of these children have this risk. So we want to come up with this multi-scale model where we know the enzymatic activity, what it does in the tissue, how the cells respond to the different biochemical, biomechanical insults to produce these enzymes that changes tissue structure, and then hopefully come up even with some biomarkers that we can test from serum that we can then put into some computational predictive systems to say this child might be at risk, even earlier on before they start having the noticeable strokes. And so I also got to talk about our cardiovascular and HIV-1 work. This is actually Francois Barry-Sanoussi, who won the Nobel in 2008 for identifying HIV as a cause for AIDS. And so this was something I went to my first summer here. It was, NIH was offering this scholarship to introduce new investigators to HIV research. If you had never done it before, they were having this program to teach us all about it. So it was in Cape Town, South Africa, so clearly I wanted to go. And I applied and I got accepted, which was great. This is Cape Town from the back, or from the far side. But there's a group of us here. But what it started to really show me is that there are a lot of things they still don't know about HIV, okay? And the biggest problem is that people with HIV are living longer, but they're dying of all these other diseases. So we all used to think about, oh, you gotta protect T-cell count, et cetera. So the drugs are really good at suppressing the viral loads and raising CD4 T-cell counts, but they're dying of things like heart attacks and strokes. So they did this study called the SMART study, where they thought that the drug was causing the problem. So they put people on intermittent drug therapies, and thinking that they say the amount of drug you were exposed to, they would live longer. The people on that intermittent treatment actually died more than those that stayed on the study drug regimen. And this kind of gave the clue, well, there's something the virus is doing and there's something the drugs are doing. So it's this combination effect. And they have other studies that failed, but as I was at this meeting, this conference, it was one of these things of, they don't have a good system to study. So HIV is a human virus. It only infects, well, not human virus, but it only infects human cells. So we've all heard about the cat model and the monkey model, but those are different viruses that infect those cells. So all of the HIV human cardiovascular work was done with patients, all on different regimens, all eating different things, all at different places in the world. And so, the biostatistics were just trying to normalize for different controlling factors, confusing it. So I came back and started to partner with Rudy Gleason here in biomedical engineering and mechanical engineering, and also with Roy Sutliff, who's at the Emory VA, to try to come up with some better ways that we could study this. So Roy actually has this HIV-1 transgenic mouse that actually expresses not the virus, but all of the proteins that the virus makes. Because there's been some literature that supports that just some of these viral some of these viral proteins actually can induce cardiovascular damage. So Yvonne Kennedy in my lab came on board and started to look at this. And again, we also care about how that relates to directions of flow. And so in the greater curvature of the aorta, this is exposed to the high atheroprotective flow. And in the lesser curvature, you get this disturbed flow atherogenic profile. So she collects this for histology. And then she also collects the thoracic aorta, which has fairly unidirectional flow, and compares that to what's happening at the abdominal aorta, where you get regions of flow reversal, and comparing how does flow reversal or normal flow induce cathepsin activity, and then how is that different in the presence of these HIV proteins by comparing these to a wild-type mouse. And for the sake of time, I'm not going to show any results on this. I just wanted to generate some thought, enthusiasm, and if anyone wants to work with us on it, we always are welcome to more people. But to get to that human problem, at Georgia Tech, we're, the kings and queens of tissue engineering. And so Rudy Gleason had been working with Bob Nerum to develop the human tissue engineered artery with Julia Rakin and his group. So we can make a human artery that is comprised of human cells that can be infected by this human virus. And what we can then do is look at what role is that human virus having on human cells to cause cardiovascular dysfunction. And so we're using these three-dimensional systems. This is actually a collagen disc, just to show that when the monocytes do bind, we can see them with endothelium, and monocytes will penetrate into the depth and differentiate into macrophages, because again, the monocytes are what are infectable. Endothelial cells are not. And then, of course, the beauty of it is that Rudy has this great bioreactor that we can cannulate our vessels to, and we can actually put through physiological wave profiles or fluid profiles to then say, under this environment with disturbed flow or protected flow, and the presence of this virus being released by these cells, what sorts of human physiology are we seeing? And so this is great. We got funded for the grant, which is exciting, and I got to go to Vienna. And it's a very activist organization as well, so I'm in a march here, actually. But the reason I also wanted to show this is for all the new investigators out there. They now have a new call out. They're taking a second class because we were so awesome. And proposals are due October 17th. Again, new investigators that have never studied HIV before. They want people who bring fresh ideas into studying either the virus itself or any of the comorbidities. So it's $150,000 for 2 years. Very great group. Next year the conference is in DC, which seems boring, but it's really exciting because it hasn't been DC in 15 years because of US laws. But the year after that is Kuala Lumpur, Malaysia, and they will fly you out there. So definitely apply, and I'm willing to talk to anybody about that grant. And I guess my final thing we're going to talk about is cancer, right? Cancer's huge, and actually cathepsins play a major role in cancer and many different types of cells involved in the cancer. So So we used our zymography since we could detect multiple enzymes in there, and we actually found that in doing this with human breast cancer samples that we purchased, using normal tumor, normal tumor, normal tumor, you can see off in the normal tissue, on in the tumor, okay? Particularly for Cathepsin K. Cathepsin S has some shadows and L, but K was often normal on in the tumor. 50-fold increase, and then Cathepsin L had a 9-fold increase. Now, whenever I talk about cathepsins to anybody, the biggest question, what about MMPs? What about MMPs? So we look at MMPs, okay? MMPs, you can see they're up in normal, up in tumor. They serve many different constitutive functions in many cells, as we know. And so they were increased, but only by 2 to 3-fold in the breast cancer. So compared 2 to 3-fold with 50-fold, we look like we got a really good biomarker. So the question is, the zymography is actually very cheap. There's no antibodies used. We're using 10 micrograms of protein. I mean, we're using nothing. And since it's seeing this off-on signal in the breast cancer, is there a potential use for a clinical diagnostic? And so what we then did is we had 10 patients. We took their normal and tumors. I'm only showing 5 here for the sake of space. Again, normal tumor, normal tumor, normal tumor, normal tumor, normal tumor, and compared it for K and for L. But if you actually break it all the way apart, you can see here that there is almost a threshold that you could draw. My hand is shaking, so I can't do it all the way good. But a threshold you could draw below which is normal and above which is tumor. So can you have some absolute threshold value that you can determine? And the way we determine this absolute amount is, you can see, we put in a standard curve of enzyme as we loaded the gel. L was not as consistent because you have these two that are actually quite low, and then a few are actually higher. And again, when we looked at the MMPs again, you know, they're all over the place. So you couldn't just say one way yes or no, just looking at MMP activity. And so Ben Chin, great undergrad of my group, what he did, he's actually the one that's been working on this. And so he then took the data and did the sensitivity and specificity analysis. And just based on those 10 patients we had, again, 10 is a small number, but we actually were able to get 100% sensitivity and specificity for Cathepsin K, and only Cathepsin K. With L, we got 100% specificity, but only about 80% sensitivity. But again, we calculate this window of opportunity where if we were to draw this threshold right in the middle, I mean, you could almost have this off-on binary yes or no from a breast tumor sample. Now, again, the problem still it is from the actual tumor. So working with an oncologist, you know, the idea would be that fine needle aspirates is what we could then use to capture that tissue and then the process. And even better, this assay right now, when we started, it took 24 hours to get a result. And as you know, currently they have to send it to the pathologist. There's some processing that has to happen. You got to get the time of the pathologist. And then they look at it by eye and say yes or no. This would be quantitative, number one. And my postdoc, Gerald, has been working to minimize the time it takes. And he's got this assay down now to about 4 hours of saying yes or no to a breast cancer diagnosis. And where we're looking to go with this is actually, of course, to sample the tumor, where we can then quantify how much activity we have with zymography. A student is working on this in situ zymography to get the location of the proteolysis, and then match that into our kinetic models to get a prediction of how bad is your tumor. How much matrix will this thing turn over before it starts to get away? So you can get a more personalized idea of how active your own tumor is. We'll skip past this first now. And then, good, I got 5 minutes to do this one. Good. Because this is the new stuff we got also. So I love all the rest of it. But again, to get that true predictive part of the cancer diagnosis we just discussed, we had to do a full model construction. We had it just for K, but again, as I showed you, KLS, I have B here. B was an old favorite, but everybody studies B, so we kind of moved on. This is not going to be V. But they all can hydrolyze the same substrates with different affinities and different catalytic rates. But they also bind to the inhibitor, different affinities, different strengths. And they can all turn over protein in different ways. So if we're looking at what's happening in a cell or a tissue, we want to be able to account for all of this. No easy feat. But so Zach and I decided, well, let's take a first step. All the biochemists have done one enzyme at a time and done this great characterization of that one enzyme, and it's brilliant, right? So we're like, well, okay, let's just start and put two together, because we haven't seen it yet, and we've looked through the literature. So what always happens is, these two are not together yet, but what always happens is when we run these fluorescent assays using a fluorescently labeled elastin or collagen, this is actually elastin, you start to lose activity around 2 hours. It always just plateaus. And you talk to I went to the Gordon conference and talked to the people like, what's happening? They just stopped working. Well, what's happening? They just stopped working, right? Not a good answer, clearly. And so what we did is we ran blots after it, and we actually found these enzymes were degraded. They were gone by the end of this period. So where you start to see this level off, it's not that they've stopped working, they're done for, right? So this was our first kind of exciting thing. We found, we're like, oh, okay, so we need to think about this. Well, let's put the two together, and maybe we'll get this combined activity. because in a system, that's really what's happening. So using, so the open triangles here are from an actual elastase experiment. And then the red line here is using our model of if you combine the two in one tube towards the same substrate, this is how much elastase activity you'd expect to get. But we were only getting this amount, the red circle, which again, puzzled us for a very long time. Not a very long time, but it puzzled us. But then when we actually ran the blots, it turned out S was staying around a lot longer, but K was being degraded a lot faster when we would put the two together. What? So, and this is in the presence of elastin. So then we used our zymography, and again, the beauty of zymography, you get both enzymes on the same gel from the same sample. And here you can see the K is actually gone while the S signal is still present. So we said, well, it looks like they are interacting in a way that we had not imagined. And so what we began to do is incorporate this term of cathepsin S degrading cathepsin K. And when we fit the model based on that, you can see we now have a nice fit here with the red circles. And it actually changes the contribution to that total activity that you thought was from K and that was from S. And so we then, of course, had to come up with a fancy name for it. So we call this cathepsin cannibalism, okay? And this has not been shown before, and this has been quite fantastic because, again, many of these cells release several altogether. And so a big problem with cathepsins is that when they develop these inhibitors and pharmaceutical inhibitors, there's major side effects due to dosing, major side effects. A big problem is, again, I think they can't actively get the mature form, so we're hopefully helping them that with zymography. But again, if you overdose, there's cross-reactivity with other cathepsins that serve important homeostatic functions, plus you may be overdosing and hitting other targets. So we thought about this cathepsin cannibalism. And the first studies were all done on elastin. Now remember, Cathepsin K loves collagen 1 better than any other enzyme. And so we said, well, does it still degrade or go away as fast in presence of collagen 1? And it actually didn't. Now gelatin is denatured collagen. You can see it still goes away quickly in gelatin, but collagen 1 seemed to stabilize it being around longer. And you can see below it's cathepsin S. But then when we still co-incubated it with cathepsin S, you're still getting this degradation of K. So cathepsin S cannibalism of K is still happening regardless of any enzyme stabilization effects on collagen 1. And again, you can see the same on gelatin. And what's also important here to note is these are incubated with collagen. These are SDS page run afterwards. You can see that cathepsin K degrades that collagen 1 because it does. That's what it does. Cathepsin S does not. Very poor type 1 collagenase. But they both degrade the gelatin. So we were thinking, well, if S can degrade K but not the collagen, there are many diseases where cathepsin K is up, and it shouldn't be, like osteoporosis, for example. So we went back to our model and thought, well, if we increase the ratio of S to K, is it possible that S can eat the K and preserve the collagen structure? And so Zach first put in the model looking at how much of the collagen degradation was happening, and as you increase the ratio, you get less collagen forming, and you also get less cathepsin K remaining. And this is from the computational model. And so we went and did it. And incubated, this is a Western blot. And you can see when you get to a ratio of 10-fold, you see a decrease in cathepsin K. This is after 1 hour incubation. But even better in the zymography, it captures even better because you can see both enzymes together. So you see at the 10-fold, that's where you see this loss of the K activity. And you also see where you have the greatest amount of cathepsin S, all in the signal. And then even better, when you look at the SDS page of how much collagen 1 is present, you can see that's where you get the maximally protected type 1 collagen. So this was really an exciting finding. Instead of trying to use small molecule inhibitors, perhaps in the right context, you can use one enzyme to degrade another. And what was brilliant, there's actually really nice things Zach had to do to actually get a better fit. The other model really didn't fit well. What we had to think about is type 1 collagen can be degraded by K, but it can't degrade S. But when K first cleaves it, you now produce products that S can hydrolyze. And so it kind of adds this nonlinearity. And when adding in these terms, we were actually able to get a much better fit of what we were seeing as we increased that S to K ratio. So this is what we're writing up now. We're very excited about this whole idea of cannibalism. And so just in the length of time, would like to thank my entire group. It's been a great 2.5 years. Look forward to 20 more. I'd like to thank funding, the innovator, Georgia Foundation of Georgia. We couldn't do it without the sickle blood. These are some of my early sickle collaborators. International AIDS Society, Rudy Gleason, Roy Sutliff, Laura Hanson, Julie Rankin work on this project with my group. And then the Georgia Cancer Coalition, and John Kennedy is the oncologist that I mentioned. And of course, GTEC gave us money for some of our monocyte work. And these are people who are either not in the picture or aren't in the lab anymore that were very instrumental in helping us get going. And so we're here to fight disease and health disparities. And since 2009, we're going to keep going. So thank you for your time, and I'm open to any questions. Oh, yeah.
Very nice talk.
Thank you.
And I had a question with when you were able to show, that there was maybe a possible drug therapy, using the JNK. You were looking at all those different MAP kinases, and you show, for example, that nothing happened with ERK.
Yeah.
And as you said, ERK is usually involved with everything.
Everything, yeah.
But at the same, in general, all MAP kinases are involved in a lot of things.
Right.
So do you think that targeting, JNK, for example, could have some other, implications when you, downregulate that one, if you try to degrade systemically? Or?
Yes, and yes, the answer is yes, we do. But what we have to think about with these individuals is this balance of, the good versus the bad. Because right now their life expectancy for a man is like 36, 42 for a woman. And so if you can reduce some of the, JNK is one of these, stress kinases. But right, if you guys have, we are open to ideas about targeting, but we actually don't know where you need to target it just yet. But right, these are things we have to consider. Because JNK is one of these ubiquitous things also, and you don't want them to be open to other diseases because we've knocked down some of their immune responses. Good question though, yeah. Yeah.
What is the role of Cathepsin K in the metastasis, and is it involved in the EMT process?
So yeah, Cathepsin K is very interesting. In the metastasis process, it helps to eat through the collagen around it and surrounding tissue. Then when it gets to the bone, there's actually this feedback between the tumor cells and the osteoclast. So the tumor cells spit out rank ligand, which activates the osteoclast to pump out more K. Then K degrades the matrix, which releases growth factors bound in the matrix that feeds the tumor cells. And that's kind of the general concept of what's happening here. But then also people have shown Cathepsin K can also degrade certain cell receptors. I know it degrades CXCR4 and kind of stops that SDF1 signaling from happening. And I think Cathepsin S or B does actually degrade some of the cadherins that promotes EMT as well, and other cancer. I've seen that in ovarian cancer models. Yeah. Yeah.
So in these sort of extracellular, if I understand correctly, or non-lysosomal activities for cathepsin, do you think it's actually the proteolytic activity, or do you think it's some sort of signaling cascade that's actually involved?
Beautiful question. I did not plant that question. So the way that I always think about it is I think about the cell in context. And so I think when the cell degrades matrix, cells get different signals from degraded matrix than they get from intact matrix. Or if they lose one type of matrix protein, then they lose some of that signaling. And there's also shown that when the matrix gets degraded by certain cathepsins, it releases bioactive factors like collagen 18, for example. When L cleaves, it releases endostatin that prevents that's vasoconstrictor. So there are things like that that happen. So I think it's linked, where it changes the environment and then the cell senses that and then they change.
I think it's cathepsin cleaving at pH 7 or something like that.
Right. Yeah.
As opposed to I was thinking more like it's in an inactive form, but the interactions it has on the surface, in other places, non-activity might be affecting some sort of signaling cascade that you're just not knowing yet.
And that could be true. What we're also seeing, though, in some of these studies is that there is some allosteric binding to different proteins, and then that whole thing is we don't know what could be happening. But that could also very much be true, yeah. Okay, thank you very much. We're around.